Hi, my name is Rhiannon and I'm a third year PhD student at the University of Leeds, funded by Brain Research UK. My research focuses on a type of brain tumour called glioblastoma, which is the most common and aggressive form of adult brain cancer. Despite patients receiving treatment consisting of surgery followed by radiotherapy and chemotherapy, almost 100% of the tumours regrow. This is likely due to some of the cells that are left after surgery being treatment resistant so that they survive and regrow to form a recurrent tumour. We have identified a gene that is present at consistently higher levels in recurrent tumours, meaning that it may be conferring treatment resistance to cells in which it is switched on. This gene is called SLC6A6, and the aim of my project is to find out more about what it is and see if it is contributing to treatment resistance in glioblastoma. To determine if SLC6A6 is important, I am treating cells taken from patient glioblastoma samples and grown in the lab with chemotherapy and radiotherapy and observing how they respond to treatment. Following this, I will use techniques to switch the gene SLC6A6 off in some of these samples to see if it makes them better or worse at surviving treatment. If a gene is important, removing it should make the cells less likely to survive treatment. I grow the cells I work with as miniature tumours, known as spheroids, to mimic the 3D properties of an actual GBM tumour that grows in the brain. So far, I have managed to optimise growing spheroids, and I have developed a new automated way to record their growth over time so that I can see how it differs when we manipulate our gene of interest. Recording the growth patterns of around 100 miniature tumours used to take 2 hours, but now it only takes 2 minutes, giving me a lot more time for experiments. I have used biological methods and identified inhibitors that one day might form the basis of actual drugs that switch off this gene in GBM cells. I am now in the process of applying some of the same treatments that GBM patients receive and seeing if switching off the gene makes the treatments more effective. If our findings prove that SLC6A6 is related to treatment resistance in glioblastoma, we could then investigate how targeting the cells expressing this particular gene could create a more targeted treatment for glioblastoma patients. Ideally, we would then target these cells alongside the current treatment to kill more of a tumour and stop it from growing back. Thank you for listening to my research. I hope you've enjoyed it.